Thanks everyone for joining. Thanks for joining the webinar today. So the webinar that we're going to be going over is related to Post-Translational Modification Analysis with PEAKS Studio 8.5. All right, so I'm going to start off the presentation talking about a little bit of the goals and challenges of analyzing PTMs with mass spectrometry. So the problem with identifying modified peptides is that they're often low abundance in comparison to their unmodified counterparts. Yeah, so often the case is that you'll enrich to wash away the unmodified peptides and focus in just on those modified peptides. PEAKS is able to work with that pretty well, but we also have been shown to work pretty well at identifying the low abundance modified peptides too, and I'll show that in a couple examples. All right, another big problem with identification when it comes to modified peptides is localizing the modification. The reason being is often the case that you have a few different amino acids within a peptide sequence where the PTM can actually attach. So we need some sort of statistic and some methods to actually place a PTM at the position that it's being shown. So this can be done with fragmentation types. So we have a few different fragmentation types that are supported by PEAKS. So PEAKS works really well with CID, HCD, ETD, and ETHCD options. ETHCD is a new option that we added in 8.5. So ETHCD gives you B and Y ion series as well as C and Z ion series. So it's really good at giving a very rich spectrum, which is great for PTM identification. Okay, so one other thing that you often have to look at is actually quantifying similarities and differences between samples to actually see what's going on in terms of how much, for example, phosphorylation is occurring in one state versus another, as well as this could be used for stoichiometry work. Okay, so just to give a bit of an outline of what we're going to be talking about today. Okay, so we're going to be talking about in the first section about some of the common things that you need to watch out for when you're actually validating your modification assignments. So this can all be done with your standard PEAKS-DB search. And then next, I'm going to talk about a tool that we have within PEAKS called PEAKS PTM. And what this allows you to do is allows you to search for unspecified and hidden modifications. So what it does is it takes advantage of de novo sequencing as well as database search to limit your search space and allow you to search for as many PTMs as possible. The next thing I'm going to talk about is quantifying some quantitative analysis when it comes to PTMs. So we have this new tool within PEAKS called PTM Profiling. So I'll go over how that can be used to help you find out the similarities and differences in PTM amount between samples. And then I'll talk about some of the new features in 8.5, such as our updated PTM database and some advanced PTM profiling within quantification results. All right, so I'm going to be using a few examples within this talk here. So the first one is just going to be a basic comparison looking at monoclonal antibodies. In this case, just looking at an untreated antibody sample versus an antibody sample that is treated with hydrogen peroxide. To just look at the effect of hydrogen peroxide on the oxidation within that antibody. And then next I'm going to talk about a few examples related to phosphorylation. In one case, looking at enriched samples, and then as well looking at the example of low abundance modified peptides. All right, so let's look at this first example here. So here we have a breakdown of the experiment. So you have your typical approach where you just have your monoclonal antibody, you do a trypsin digest, run it through the mass spec, and then you just have the standard approach with the added hydrogen peroxide here to induce some oxidation. so how we hope that you set this up in PEAKS is that you add a different sample for each of the data files that you bring into PEAKS. So you have your treated antibody, and then you have the untreated antibody. So in PEAKS, what you want to do is you go up to the top left and go File, New Project, add your data, and if you do have multiple samples, you want to make sure that you add each of those samples from your experiment to a new sample within the software. So the reason why I bring that up is that PEAKS has some built-in tools that help you get some information out of the data that is specific to each individual sample. So if you want to get a breakdown of what's going on in sample 1 versus sample 2, you want to make sure that you load those into 2 different samples. And then as well, let's say, for example, you have multiple fractions per sample, you want to make sure that you add each fraction, add the fractions to the same sample so that they'll be grouped together in the search. All right, so here's the PEAKS-DB search parameters here, just your standard parameters that you'd get for an Orbitrap. In this case, we're just casting a bit of a wider net with a wider precursor error tolerance and including some oxidation deamination here in your search parameters. So pretty straightforward parameters here. All right, so let's talk about how modified peptides appear within the software. So here you have the protein tab, which appears in any identification result of PEAKS. And what you see here is you have a modification assignment. So how these modification assignments appear, in this case you have oxidation at this position because you have an O with a blue background. And if you look over here in the legend on the right-hand side, you can see the details of the modification and what letter and color they're assigned to. All right. So when you click on your identifications in PEAKS, it will give you an interactive display of the actual peptide spectrum match. And looking at the differences between your oxidized methionine peptide here versus your unmodified peptide, you can see as you scroll over the peptide sequence within the software, it will actually show you the fragment ions that are associated with which amino acid. So in this case here, we have your oxidized methionine shown with an M in lowercase, meaning that that one's modified, and then if it appears in uppercase, it means that it's unmodified, shown here in the unmodified spectrum. And then you can see how we're certain that we have an oxidation at this site because you have good fragmentation between your Y3 and your Y4 with this slightly different mass shift than the unmodified peptide. Yeah, so that was just a quick overview of how to interpret your basic identification results with a modification in PEAKS. Let's look at a bit more of a complicated example here. So in this case, looking at an enriched example here. So in this case, we're including the extra step of enriching for phosphopeptides using the titanium MAC beads here. So, and as well, we're also including ETHCD fragmentation to get a more rich spectrum to find our B and Y ion fragments as well as our C and Z ion fragments. Yep, and here's the search parameters here. So we're just added the phosphorylation and how these results end up appearing in PEAKS. So here we have a modified peptide assignment in the peptide table of the software. So what you'll get is you'll get your -10 log P score showing the confidence in your peptide assignment within the context of the whole database search. So we have 2 scores for a modified assignment. So you have your -10 log P, which is a score that relates to the whole peptide sequence, as well as you have A scores for each of the different modification assignments. So in this case, we have a phosphorylation at the S at position 6, so this one here, as well as we have a phosphorylation at position 9 shown here. And if you look here, we have an A score reported for each. So generally speaking, just to show if it's too small on your screen, sorry about that. So the A score for the modification at position 6 is 30, and the A score for the modification at position 9 is 45. And those are good scores because we use minus 10 log P scale for our A scores as well as our significance values. What that means is that if you have a minus 10 log P of 20 or greater, it equals a p-value of 0.01 or less. So what you're usually looking for with a p-value in this case is it's the probability that the modification is occurring at the position reported as opposed to the other possibilities. To put it in a more simple way, generally speaking, it means that you have a less than 1% chance that the modification is being reported at that position by mistake. So if you have an A score of 20 or greater, that means that you're pretty confident in that modification site. And that means that in this case we have a peptide with 2 phosphorylations on it. And you can see why we're confident in this modification here because you have your C ion series as shown here in the ion match table with the blue masses, meaning that we have matched ions for these fragments, showing fragmentation before and after our phosphorylation sites. And because we're working with ETHCD, you also have fragment ions from your Z and your C and Z as well as your B and Y, giving you extra support for those modification sites. All right, so that was just showing an identification using an enriched sample. Let's look at the more difficult case. So how does PEAKS perform when you have an unenriched sample? So looking at modified peptides in a complex background. So in 2012, there was an IPRG study that looked specifically at modified peptide identifications. So what this included is it included some low-abundance synthetic peptides with some known PTMs. And you take those synthetic peptides that are modified and you put them into a full yeast cell lysate in order to make it more complicated to identify those peptides. And then compare all the software to see how they do in terms of identifying those modified peptides within the complex background, as well as how many identifications you're able to make. So how PEAKS did, so this is just showing off how PEAKS was able to do in this case. So showing here in the plot on the x-axis, we have the number of correctly identified peptides, and on the y-axis showing the correctly identified spiked-in peptides with the PTMs. So generally speaking, PEAKS did really well when you consider both the correctly identified peptides and the correctly identified PTMs. It was able to perform pretty well in comparison to the other tools, so we're just highlighting the PEAKS identifications here. So just to go back to how we validate these results, in more recent versions of PEAKS, you also have this area column in your peptide table. So what this area column is it's the area under the curve of the peptide feature associated with your identification. So the area of this identification is 1.94 E5, which generally speaking within this experiment is a pretty low abundance peptide, but we're still able to correctly identify this modified peptide here, again with 2 phosphorylations, one at the 3rd amino acid and one at the 7th amino acid. And we can be pretty confident in this identification based on the fragment ion matches here. And how do we know that this is the correct identification? Well, in this case, it's a known peptide, so we know what we're expecting in this case. So this leads to one of the problems in dealing with modified peptides. So within this peptide here, you have 3 different sites that can be phosphorylated. You can have the S at the 3rd position, and the 5th position, and then also the Y at the 7th position. So you need to see a fragment ion that actually picks out that peptide from the others. So how do we actually assign that phosphorylation? In this case, because we have the B3 minus the neutral loss within this spectrum here, meaning that we likely have phosphorylation at the 3rd position, and then in the other spectrum here, we have the Y4 minus the neutral loss, meaning that we likely have a phosphorylation at the 5th position. So actually in this sample, we have a mixture where we have some peptides with phosphorylation at the 5th and 7th position, and some peptides with a modification at the 3rd and the 7th position, which actually turned out to be true in the case of the study. All right, so just a bit of a review. I've already touched on this a little bit previously, but I'd just like to reiterate how PEAKS actually, you can use PEAKS to make sure that you're only reporting confidently localized modification sites. So on the right-hand side of the coverage view, I'll just jump over to the software real quick to show this. If you look on the right-hand side of the coverage view, you have this box here where you can show the confident modification sites. So the default option is to use the minimum ion intensity. So let's take a look at what that means. So for example here, so just show off a little bit about this interactive display here. If you click on a peptide, it will show you the best peptide spectrum match for that site, and you can scroll along the peptide sequence and it will show you which amino acids are associated with which fragment ions. Once you pick out your modified amino acid, which will be lowercase, you can then use your mouse to zoom in just on those amino acids, and then you can see here, we have a B11 and a B12 showing that we have fragmentation before and after this modified amino acid. And if you scroll over the amino acid, the fragment ion, you can see the actual relative intensity of these fragment ions. So this one's 14%, this one's 7%, meaning that this one is confidently localized according to this minimum ion intensity. So it requires that you have at least 5% for both. So you have to have a confident B and Y ions, not both. You can have a confident set of B ions, you have a confident set of Y ions in the case of HCD or CID. In the case of ETD, you can have a confident C or Z ion series. In the case of ETHCD, you can have any of them. So anyway, so that's one of the options, is to use the minimum ion intensity, or you can use an A-score. So that will also be based on the fragment ions, but it will actually give you a statistic that you can work with. So I'm just going back to the slides here. So you can choose either of those options here. 5% or 20 for the A-score are both good options. And then once you do that, only modifications that are within those thresholds will appear above the protein sequence. Anything that is below those thresholds will just appear below. And look, if you choose, you can also choose to make those modifications that don't pass that filter just completely not appear in the display. So if you go back to the summary view, you'll see up at the top that we also have this PTM A-score filter, or minimum ion intensity. you can choose the cutoff there, click the Apply button, and then that peptide will not appear in the display. Just a quick detail here. This peptide here that is below, say, an A-score cutoff of 20, notice here when the filter is set to 0, it appears here in the display, but then we set it to 0, that modification won't appear, but it will still be included as one of the subspectra within the unmodified peptide. But for all intents and purposes, it just won't be visible in the result. Yeah. So with all that in mind, once you use your have a firm understanding of the A-score and of the minimum ion intensities and how those can be used within the software, you can be pretty confident in any of the modification assignments that are made within PEAKS. You can use these filters to make sure that you're only reporting confidently localized PTMs. Right, so with all that in mind, we can start to look at hidden and unspecified PTMs with our PEAKS PTM tool. So here's just a quick review of the full analysis pipeline of PEAKS. So you load in your raw data, de novo sequencing is performed on all your spectra, then we do our de novo assisted PEAKS DV search, and then you can include this optional PEAKS PTM search. So by default, this PEAKS PTM search can be set up by just clicking this checkbox here in the identification search page. And when you set this checkbox, it will search using PEAKS PTM with the same parameters that you have set here that will be used for de novo sequencing and database search. Alternatively, if you actually click your identification result node up in the top left and then click the PEAKS PTM button, it will bring up a search page that is specific just to PEAKS PTM. So it will show you the default settings. So by default, it searches the 313 naturally occurring modifications within the Unimod database, or you can search with preferred modifications if you select this radio button. So just to jump back to the software, so just to show the different options here to set this up, you can just set it up from your identification search here by clicking this button, or you can grab an existing identification result, click this PEAKS PTM button, and you can set up the search from here. All right. So let's go back to the initial example that we had looking at a treated antibody versus an untreated antibody. So what happened when we searched that with the full list of PTMs included in PEAKS PTM? So what you find is you're able to find some extra modifications that you may not have considered. So you can see that this protein here turned out to be pretty heavily modified. But one extra identification that we made here, which is quite interesting, is we also found an extra modification at the position where we had an oxidation of the dethiomethylation here, which can also occur when you add hydrogen peroxide. Yeah, and just to summarize here, so when you use PEAKS PTM, you can get a bit of a cluttered display, as we saw there on the previous page. So here's just a comparison of the identifications that were made with database search as well as PEAKS PTM here. You can see that a lot more modifications were assigned, but remember that all the modifications that we have assigned here are within our A-score of 20 here. So just to look at how you can get this type of display within PEAKS here, if you choose, here's the full list of all the modifications that are assigned, and up here in the legend on the right, you can switch from this coverage display to just show this outline display, which gives you a much clearer picture of the different modifications that were assigned to your protein. All right, so now that we've talked about PEAKS PTM, you have a pretty good idea of all the different ways that you can identify modifications within PEAKS. But what about when we want to consider the differences in PTM quantity between samples? And that's when PEAKS PTM profiling comes into play. All right, so here we have your typical identification, and I just wanted to show where you can actually find PTM profiling. It often gets overlooked. It's up in the top right-hand corner of the coverage view, and you can activate the display by just clicking this button here. And I just want to point out that the modifications that will be included in PTM profiling are only the ones that pass the filter that you set here. Whether you choose minimal ion intensity or A-score, only the ones that pass the filter that you set will be included in your PTM profiling. All right, so once you click that button, this display will appear. So what this gives you is it gives you a comparison of the modified area shown in red, coming from the area under the curve of the peptide features, compared to the unmodified area shown with the blue bars. And by default, it will show the display using the log 10 of the area. And that's going to be the total area of all the peptide features of your modified compared to your unmodified peptides. So just to give you some definitions here, so the modified abundance will be the sum of the peptide abundance with the confident modification at the specific site, and the unmodified abundance will be the sum of the peptide abundance without any modification at that specific site. So the display will only include the peptides with the modification that you chose and the unmodified peptides without any other modifications at that site. So once you click on a modification position, so the annotation here is that we have the methionine at the position 254. You can see the treated area, the treated modified area versus the treated unmodified area, and so on. And they'll give you the best modified spectrum compared to the best unmodified spectrum. Then up here as well, you'll notice that we can choose to use the only fully digested peptides here, or we can choose all peptides. So you can include fully digested peptides just to give an added level of accuracy to your results. Okay, so going on to the further details here, we can look at, in this case, oxidation and deamidation. We can see the similarities and differences, what's going on with these modification sites. So we can start to see that in some cases, you get more modifications. So for example, the deamidation at this position, you get much more deamidation at this site in the treated versus your untreated sample. Oh yeah, and in this case, we have the percentage modified option here as well. So you have the option to choose the log 10 area or the percentage modified. Okay, so, and then if you want to export these results, up in the top right-hand corner of the display, you can click the Save to Text format, and it'll create a nice CSV file that will then give you the raw data, which you can then use to do some further analysis. Okay, yeah, so I just wanted to point out here again, in the export, the modified amino acid will be shown in lowercase and flanked by the 10 amino acids on the N and C terminus of that modification site. So this here, in this case, will be showing the modified peptide sequence window. Okay, so up until this point, everything that we've seen will be included in the identification package of PEAKS. So while PEAKS PTM, so PTM profiling is a quantitative tool, we still include that within the identification package of PEAKS. So we have some further PTM analysis within the Q module that you can use to do some extra analysis. And before I go into that, one of the things that I want to point out about the new version of PEAKS, PEAKS 8.5, is that we've actually updated the list of PTMs up to the Unimod database in May 2017. So some PTMs had been added or removed based on research, and we've also moved glycosylation to a separate tab. So it makes it much easier to work with here because you have your list of common PTMs, uncommon PTMs, as well as glycosylation, artificial modifications that aren't included in PEAKS PTM. And one thing I'd like to point out is that we've also removed mutations, so substitutions, insertions, and deletions are not included in PEAKS PTM. If you'd like to search for those mutations, it's best to use the spider algorithm. All right, so let's talk about PTM profiling in the context of quantification results. So here we have another example showing the project tree that would appear in the top left in this case. So we have a sample collected from cells within the interphase of the cell cycle compared to cells in the mitosis phase. And the aim of this study is to see the difference in phosphorylation profiles between interphase and mitosis. So here we have some label-free quantification results looking at a specific protein here. And if you look at the sample profile, you'll notice that there is some slightly different expression of the full protein between the two samples. So one of the reasons why we made sure to include PTM profiling within our quantification is because you need to consider what's actually going on the full protein level when you're looking at specific PTM sites, because you want to make sure that the change that you're reporting for your modification is not skewed by what's actually going on the full protein level. So here, in the Advanced PTM Profiling page, you have the option here at the top to apply protein level normalization, meaning that the protein abundance will be used to normalize the actual intensities that we're reporting for each position. Some of the other added tools that you'll have here, so you'll notice here in the bottom right, you'll see that we have the areas broken down in much more detail. So as well as being able to see the peptide areas in terms of the areas of the modified peptide and the unmodified peptides, you also need to consider the question, what if that peptide actually occurs in different charge states? So here, just going from the top level down to the bottom, we have the areas reported by position. So the actual position within the protein where that modification is occurring. And then you have all of the peptides that contain that position and with that modification. And then once you select a peptide, you'll also be able to see the different peptide features that contain that modification site as well. So this will all be summed up from the peptide feature level to the peptide level, then to the position level. So as well, we also have the extracted ion chromatograph showing the interphase and mitosis here. So to give you a quick way to actually validate the area under the curve that we're calculating, as well on the bottom left here, we have a heat map where we have the ratio shown at each of the positions, the modified abundance within a group over the average modified abundance of all samples. So what we then see here, for this protein, at most positions, we're seeing upregulation of the phosphorylation at most of the positions where phosphorylation occurred. So from positions S32 to 660, we see upregulation of phosphorylation, then a slight difference for the phosphorylation at position 14. Yeah, so that was PTM profiling within label-free quantification. I just wanted to quickly point out that you can also do this with TMT as well as SILAC quantification. So here we have a similar experiment looking at to compare phosphorylation profiles specifically of kinases in leukemia cells. So I just wanted to point out that this can also be done within SILAC. So in this case, you can also see the details here. So I just wanted to point out some of the differences here. So the heat map in this case will always be shown to your reference sample here. So you'll be able to see the positions that are upregulated and downregulated in terms of phosphorylation in comparison to your reference. And over here on the right, you can see the XIC of your light, medium, and heavy peptides features associated with these different positions where phosphorylation occurs. Okay. So, yeah, that's all that I had to talk about today. Thanks for taking the time to join this webinar. And if you have any questions, feel free to go ahead and ask them now.